ongoing topic. We were discussing on microbes as a biofertilizers. We talked about एग्रीकल्चर yield इंक्रीज क्यों करनी पड़ती है, उसकी वजह से केमिकल फर्टिलाइजर ज़्यादा से ज़्यादा चाहिए, उसकी वजह से पॉल्यूशन हो रहा है. अगर ऐसा हो रहा है तो इसके रिप्लेसमेंट में हमें अगर पॉल्यूशन को कम करना है तो क्या यूज़ करना होगा? ऑर्गेनिक फार्मिंग, यानी कि यूज़ ऑफ बायोफर्टिलाइजर्स. अगर हम बायोफर्टिलाइजर यूज़ करते हैं तो उसका डेफिनेशन क्या है? तो ऑर्गेनिजम्स which are enriching the nutrition of the soil. Indirectly they are providing growth and development to the plants. तो ऐसे organisms कौन से हैं जो plants को growth and development करवाते हैं? The examples are bacteria, algae and blue-green algae. This BGA, they are also known as Cyanobacteria, as they are photosynthetic bacteria. So they are also known as Cyanobacteria. Then हमने देखा था टू वेरी इंपॉर्टेंट एग्जाम्पल जिसका फंक्शन एक ही था, टू फिक्स नाइट्रोजन. तो फर्स्ट एग्जाम्पल वाज राइजोबियम बैक्टीरिया. राइजोबियम बैक्टीरिया वेर एबल टू फिक्स एटमॉस्फेरिक नाइट्रोजन एंड टू कन्वर्ट इट इनटू ऑर्गेनिक मैटर और ऑर्गेनिक फॉर्म, और वो ऑर्गेनिक फॉर्म कौन यूटिलाइज करेगा? बाय द रूट्स ऑफ प्लांट. दिस वर राइजोबियम बैक्टीरिया. उनका name itself, it was suggesting rhizo means roots, means they are present in the roots of the plant and they are protecting the plant from different harmful insects. Next we have seen Azotobacter and Azospiridium. उसके लिए ट्रिक याद रखनी है आवाज AB यानी कि Azobacterium and AZ यानी कि Azospiridium. उसके लिए हमने देखा था they were just fixing nitrogen and they are free living in the soil. This was all about our last lecture. We will continue our topics with the same as microbes as biofertilizers. उसके examples हम different लेंगे. यहाँ पे example है mycorrhiza का. So we will start with our today's topic. Mycorrhiza. They are of genus Glomus. हर साल एग्जाम में ये क्वेश्चन आता है. Mycorrhiza are of which genus? You have to remember it. You have to write it blindly that Mycorrhiza are of genus Glomus. कौन से genus के होते हैं? Glomus. Next. So Mycorrhiza are of genus Glomus. They have symbiotic association with fungi and plant. Symbiotic means they are having mutual relationship such that plant is providing something to fungi and fungi is giving something to plant. Now you obviously know what is fungi. They are saprophytic or they are heterotrophic in nature, means they are feeding on dead and decaying matter. Now what will fungi do? इससे पहले के दो एग्जांपल्स में हमने क्या देखा था? Rhizobium bacteria and Azotobacter and Azospiridium. They both were feeding on only nitrogen. They both were taking atmospheric nitrogen and they were converting to organic form and they were providing it to plants. तो वहाँ पे एग्जांपल था सिर्फ नाइट्रोजन का. अब यहाँ पे एग्जांपल लेंगे फास्फोरस का. कौन इसको फिक्स कर रहा है और कौन प्लांट्स को फास्फोरस प्रोवाइड कर रहा है? माइकोरिजा. माइकोरिजा दे आर हैविंग सिम्बायोटिक एसोसिएशन, यानी कि फंगी प्लस प्लांट के रूट्स, ये दोनों का जब एसोसिएशन होगा उसको बोलेंगे सिम्बायोटिक एसोसिएशन. इफ यू नो एग्जैक्टली व्हाट आर लाइकेंस, अगर आपको पता है देन दैट इज द एसोसिएशन बिटवीन algae and fungi. If algae is giving something to fungi and fungi is giving something to algae, they both are having symbiotic association and they are known as lichens. And here the symbiotic association is between fungi and plant roots. So both are different but examples are same. अगर algae और fungi दोनों association बना रहे हैं तो उनको बोलेंगे lichens. अगर fungi और plant के root को association बोल रहे हैं तो that is known as symbiotic association simply. यहाँ पे हो क्या रहा है? You can see here the plant. The plant having roots. These roots are not capable to take direct phosphorus. So what fungi will do? As they are heterotrophic or saprophytic in nature, they will feed on dead and decaying matter. And whatever phosphorus they will take, that phosphorus they will provide to the roots of the plant. अभी जितना भी phosphorus fungi produce करेंगी वो सब कौन ले लेगा? Plants. Then these plants will transfer from downward to upside to the whole parts of the plant. So Here fungi that are heterotrophic and saprophytic, they are feeding on dead and decaying matter, and they are taking phosphorus from the soil and they are providing it to the roots of the plant. This is the basic mechanism, or you can call it as symbiotic association of fungi as well as plant roots. अब दूसरा point, till here you are clear. What are the functions of mycorrhiza? अब तक क्या देखा? Mycorrhiza, they were showing symbiotic association. Symbiotic association यानी कि fungi जो सेपेरोफाइटिक और हेट्रोट्रॉपिक है वो क्या दे रहा है? डेड और डिकेइंग मैटर पे फीड कर रहा है. जो भी फॉस्फोरस सॉइल में इकट्ठा होता है विद द हेल्प ऑफ फंगी, वो प्लांट के रूट्स में से पूरे प्लांट्स में जाता है. यानी कि फंगी प्लांट्स को क्या देता है और प्लांट्स के रूट को क्या देता है? फास्फोरस. एंड इट विल ट्रांसफर टू द होल पार्ट्स ऑफ द प्लांट. दे आर ऑफ जीनस ग्लूमस. यहाँ तक हमने देखा. अब उसके फंक्शंस क्या है? हाउ दे आर प्रोटेक्टिंग प्लांट, वी विल सी. फर्स्ट फंक्शन इज दे प्रोवाइड रेजिस्टेंट टू द रूट बोर्न पैथोजन. अगर रूट्स में कोई भी पैथोजेनिक पेस्ट या इंसेक्ट प्रेजेंट है, यू कैन सी दिस प्लांट व्हाट आई हैव ड्रॉन हियर. रूट्स के इस साइड में थिक लेयर है. वो थिक लेयर कौन सा है? जहाँ पे माइकोरिजा बैठे हुए हैं. माइकोरिजा दे आर द बैक्टीरिया विच आर प्रेजेंट इन द रूट्स ऑफ द प्लांट. तो ये सारे के सारे माइकोरिजा है. सब माइकोरिजा ने रूट्स को कवर कर लिया, घेर लिया है पूरा का पूरा. अब क्या होगा? अगर पैथोजेंस आएंगे, अगर ऐसे हार्मफुल इंसेक्ट्स आएंगे, पेस्ट होंगे, लार्वा होंगे, वो रूट्स को डिस्ट्रॉय नहीं कर सकेंगे. क्यों? वो हार्मफुल पैथोजन एंटर ही नहीं हो पाएंगे. अगर एंटर होना है तो उससे पहले रूट को कौन कवर कर चुका है? माइकोरिजा. इसलिए कोई भी पैथोजन अंदर एंटर कर पाएगा नहीं. यानी कि फर्स्ट पॉइंट, दे आर प्रोवाइडिंग रेजिस्टेंट टू द रूट बोर्न पैथोजन. सेकेंड पॉइंट, दे आर प्रोवाइडिंग टॉलरेंस टू सैलिनिटी. सैलिनिटी यानी कि हाईएस्ट सॉल्ट कॉन्सेंट्रेशन. दे आर प्रोवाइडिंग टॉलरेंस. टॉलरेंट यानी कि किसी भी चीज को टॉलरेट करना. फॉर एग्जाम्पल, यू कैन टॉलरेट द एंगर टिल सम पीरियड ऑफ टाइम, देन यू कैन नॉट टॉलरेट, देन यू गेट इरिटेटेड मोर एंड मोर. उसी तरह से अगर सॉल्ट कंसंट्रेशन रूट्स में थोड़ा अमाउंट में है तो रूट उसको टॉलरेट कर सकता है. लेकिन अगर वो सॉल्ट कंसंट्रेशन इंक्रीज होता जाता है तो वो टॉलरेट नहीं कर सकते हैं. उससे प्लांट के ग्रोथ और डेवलपमेंट को इफेक्ट होती है. पर यहाँ पे कौन प्रेजेंट है ऑलरेडी? रूट्स के आजू बाजू में किसने घेरा कर लिया है? किसने घेर लिया है रूट्स को? माइकोरिजा ने. अगर माइकोरिजा रूट्स के आजू बाजू में प्रेजेंट है, यानी कि अगर सॉल्ट कॉन्सेंट्रेशन इंक्रीज हो भी रहा है लेकिन माइकोरिजा के होते हुए वो अभी रूट्स में एंटर हो पाएगा नहीं, तो ऑब्वियस है माइकोरिजा सॉल्ट कॉन्सेंट्रेशन से रूट्स को बचाता है. अगर सॉल्ट कॉन्सेंट्रेशन हाई हो जाए तो माइकोरिजा क्या करेगा? उसको अंदर आने नहीं देगा. सो सेकंड पॉइंट, माइकोरिजा प्रोवाइड टॉलरेंस टू हाईएस्ट सॉल्ट कॉन्सेंट्रेशन, जिसको क्या बोला जाता है? सैलिनिटी. थर्ड पॉइंट, इट हेल्प्स इन ड्रोट. यानी कि अगर पानी नहीं है, ड्राइनेस हो गया है, ड्रॉट आ गया है, रूट्स को क्या चाहिए? ज्यादा से ज्यादा पानी. माइकोरिजा क्या करेगा? सॉइल में से पानी को कंज्यूम करके रखेगा, बचा के रखेगा. और जब भी ड्रॉट का टाइम आएगा, यानी कि जब भी सॉइल में पानी नहीं होगा, पूरा सॉइल ड्राई हो गया होगा, तभी माइकोरिजा वो बचाए हुए पानी को कहाँ पे देगा? रूट्स में से पूरे प्लांट को ट्रांसफर करेगा. पूरे प्लांट को ट्रांसफर करने के लिए फिर किसका काम आता है? देयर इज अ फंक्शन ऑफ जाइलम दैट इज प्रोवाइडिंग वाटर टू द प्लांट्स. ये आपको पता होगा, जाइलम एंड फ्लोएम. जाइलम इज ट्रांसफरिंग वाटर, फ्लोएम इज ट्रांसफरिंग शुगर, दैट इज फूड. तो माइकोरिजा विल सेव सम अमाउंट ऑफ वाटर सच दैट जब ड्राइनेस का टाइम आएगा तभी प्लांट को क्या चाहिए होगा? वाटर. तो तीसरा फंक्शन क्या है? दे हेल्प इन ड्रॉट, दैट इज dryness. Now, then they are of genus Glomus, and at last they provide increased plant growth and development. सभी चीजें देखी हमने. देखा माइकोरिजा क्या है, कौन से जीनस के हैं, वो कैसे प्लांट को हेल्प करते हैं, क्या प्रोवाइड करते हैं, हमने देखा. दे आर ऑफ जीनस ग्लोमस. दे शो symbiotic association, that is fungi plus plant roots. They are present in the root of the plant. They feed on dead and decaying matter. They are heterotrophic. They are saprophytic. They provide phosphorus to the plant root such that plants get phosphorus in enough amount. Next point we have seen that they are having three functions. They provide resistance to the root-borne pathogen. Pathogens roots में नहीं जा पाएंगे. Pathogens नहीं जा पाएंगे तो roots डैमेज भी नहीं होंगे और plant का ग्रोथ इजीली होगा. टॉलरेंस, किससे टॉलरेंस करवाते हैं? सैलिनिटी, यानी कि अगर कॉन्सेंट्रेशन सोल्ट का बढ़ गया है रूट के बाहर तो रूट के अंदर वो फिर भी नहीं जा पाएगा क्योंकि यहाँ पे कौन प्रेजेंट है? माइकोरिजा. माइकोरिजा यहाँ पे रूट्स की रक्षा कर रहा है, वो उसको घेर रहा है. अगर उसने वहाँ पे अपना हैबिटेट बना लिया है रूट्स के आजू बाजू में तो वो सॉइल को एंटर होने देगा नहीं. एंड दे हेल्प ड्यूरिंग द ड्रॉट टाइम. अगर ड्राइनेस है तो वो प्लांट को पानी की सप्लाई करेगा थ्रू द जाइलम, पूरे प्लांट में ट्रांसफर होगा. तो एट लास्ट समरी क्या हुई? Mycorrhiza are allowing proper growth and development to the plants. So it is the last point. They provide increased plant growth and development. This was all about Mycorrhiza. They are of which genus? What they provide to the plant? Very important. Phosphorus. कौन सा जीनस है? ये भी बहुत important है. They are of genus Glomus. उनके 3 functions जो बहुत ही important है आपको याद रखने हैं और वो कहाँ पे प्रेजेंट होता है? इन द रूट्स ऑफ द प्लांट. I hope today's lecture about mycorrhiza, that are microbes as biofertilizer, it is well understood to you all. Stay tuned for the next lecture. Thank you so much.
Discover the value of mycorrhizae, tiny soil-borne beneficial fungi that have a huge impact on plant health and yield. Mycorrhizae have evolved over hundreds of millions of years to help plants collect water and nutrients. Approximately 95% of plant species benefit from this symbiosis. They connect to plant roots and grow into branching root-like structures called hyphae. Mycorrhizae multiply by propagules, which are spores and root fragments containing hyphae and other fungal structures. They can be applied to the soil by drench, soil incorporation, seed treatment, soil injection, in furrow, or any methods that can put the propagules in contact with growing roots. Plant roots diffuse exudates into the soil and trigger a germination response from mycorrhizal propagules. These begin to develop into hyphae that penetrate plant roots, establishing nutrient and water transfer mechanisms. The hyphae function as an extension of the plant's root system. As nutrients are depleted around the plant, hyphae continue to grow into the surrounding soil. They absorb and deliver micronutrients, macronutrients, and water to the plant that are otherwise inaccessible by the plant's roots alone. In return, the mycorrhizae receive carbon in the form of sugars and lipids from the plant produced by photosynthesis. Mycorrhizae help the plant in other ways too. They secrete enzymes that can break down tightly bound nutrients and make them accessible to the plant. Hyphae can absorb nutrients and water along their entire length, while plant roots can only absorb through their root tips. Mycorrhizae are the only soil microorganism that produces glomalin, a glycoprotein that binds soil particles into stable aggregates, improving soil structure and stability, and increasing water infiltration, water holding capacity, and carbon content within the soil. The direct and indirect benefits that mycorrhizae provide to plants ultimately improve plant vigor, promote better flowering and fruiting, and increase yield. Plants with a mycorrhizal relationship are also more resilient to environmental stress, such as drought and temperature extremes. Benefits of using myco-apply inoculum include a diversity of species that provide different benefits under various circumstances, multiple formulations for a variety of crop needs. They're all natural and sustainable, and many products are OMRI listed. Find out how tiny microorganisms can help your bottom line in a big way. Visit mycorrhizae.com today.
You may have heard the term mycorrhizal fungi, sometimes called mycos, in recent years, but what exactly are they? The origins of the word offer the simplest explanation. Myco comes from the Latin word for fungus, and rhiza is Greek for root. These fungus roots can be greatly beneficial to gardeners. Mycorrhizal fungi belong to the same group as mushrooms, the Fungus Kingdom. A mushroom is similar to an apple. It is the fruiting body of a larger organism. But, rather than growing from a tree, the mushroom is formed from an interconnected network of root-like tubes called hyphae. Most hyphae are very small, between 1/10 to 1/50th of the width of a human hair, making many of them invisible to the human eye. More than 450 million years ago, certain types of plants and fungi formed a symbiotic, or mutually beneficial, relationship with each other. The plants give the fungi sugars that they produce through photosynthesis. In exchange, the fungi give the plants nutrients and water that they extract from the soil. 80 to 95% of land plants benefit from some type of mycorrhizal relationship. What does this mean for gardeners? Let's say you plant a seed in soil without any mycorrhizal fungi. The plant's roots will go out in search of nutrients and water to feed its growth. As the available resources are used up in one area, the roots grow further and further out, expending more and more of the plant's energy. Now, let's introduce some mycorrhizal spores. The plant sends signals out to any mycorrhizal fungi in the area, and the spores respond by growing hyphae in the direction of the signal. If a spore is able to find the root of the plant, the hyphae will penetrate the root and begin to grow structures that allow for the plant and the fungi to exchange resources. The mycorrhizal fungi will now begin to grow an extensive cotton ball-like mass that will give the plant access not only to the nutrients and water outside the reach of its roots, but, since the hyphae are so small, they can access nutrients in the small spaces between the roots as well. Though most commonly grown plants will benefit from mycorrhizal applications, there are a few exceptions. The Brassica family, which includes plants such as broccoli, cabbage, radishes, and kale, is one. Beets, chard, blueberries, cranberries, and rhododendron are some others. Though mycorrhizal fungi are naturally occurring in most soils, the application of fungicides and pesticides, the use of heavy machinery, and leaving soils without any plant growth for weeks at a time have created soils with little or no mycorrhizal fungi populations. The ground surrounding newly built residential neighborhoods and commercial properties is particularly lacking. Only in recent years have commercial mixes become available to reintroduce these important organisms back into garden soils.
Hello students, I hope you all are doing well. In the last lecture we discussed about microbes as a biofertilizers. In that we have seen example of Cyanobacteria. Today's lecture we are going to see about Cyanobacteria. In the last lecture we discussed about Mycorrhiza. Back to that we discussed about Rhizobium Bacteria. We have seen what are rhizomes. They are staying in root and they are helping plants by catching atmospheric nitrogen and converting it into organic form. That organic form will be utilized by the plant. As plants are not able to take organic nitrogen in the form of from the air, so they are using Rhizobium bacteria such that they can synthesize that nitrogen in the form of organic form. Then we have discussed about Azobacterium and Azospirillum. Both of them were responsible to fix nitrogen and they were free living organisms in the soil. Then we have discussed about particular organisms which are staying in the soil and how they are acting as a symbiotic relationship. the example of symbiotic, or you can call it as a mutual relationship, that was we have seen for lichens, that are algae and fungi. With that we have seen very basic example, that was fungi and plants. Fungi which are staying in the root of plants, exactly they are depending on dead and decaying matter. After taking dead and decaying matter, they will provide phosphorus to the plants. So it is a mutual relationship between plants and fungi, which was shown with the help of Mycorrhiza, and they are of genus Glomus. That was very important point. Then we have seen three functions of Mycorrhiza. First was they were providing plants to be resistant from pathogenic organisms. Second, अगर salinity यानी कि अगर salt का concentration बढ़ जाता है plants के roots के area में, then they won't be able to go inside. Why? Because already mycorrhiza has taken place around the roots. Third very important function was, whenever drought is taking place, that is dryness around the soil, dryness in the soil, there is no water present, what mycorrhiza will do, they will store somewhat amount of water in them and they will provide it to the plants. Then plants will be using xylem to pass it throughout the plant body. This we have seen in the last lecture. Today we are going to discuss very important point, that is known as cyanobacteria. As they are photosynthetic in nature, so they are known as cyanobacteria. In the last lecture we have seen BGA, that was blue-green algae. This blue-green algae, it is also known as cyanobacteria. So today that is our main topic. Cyanobacteria, they are the organisms that are used as biofertilizer. Cyanobacteria are also known as BGA, that is blue-green algae. They are autotrophic in nature. Autotrophic means they can synthesize their food by their own with the help of sunlight. As they are photosynthetic, they are autotrophic too. And they can fix atmospheric nitrogen. Now you have to remember here Rhizobium bacteria. The same function was seen in Rhizobium bacteria. Rhizo means staying in roots. They were able to fix atmospheric nitrogen into organic form. In the similar manner, cyanobacteria, which are autotrophic in nature, they will also fix atmospheric nitrogen. Examples of cyanobacteria are Anabena, Nostoc and Oscillatoria. Examples are Anabena, Nostoc and Oscillatoria. To remember this example, you have to remember one trick, that is an NOC. This N, it means it is Anabena, N means Nostoc and O means Oscillatoria. This C is responsible to remember or to make you remember that it is an example of cyanobacteria. ये किसके examples हैं? They are the examples of cyanobacteria. So C is reminding you, these three examples are of cyanobacteria: Anabena, Nostoc and Oscillatoria. If you can remember like this, it's better. Next point, cyanobacteria, they are present mostly in paddy fields. They are present where? In the paddy fields. And next point, they add organic matter such that they are adding organic matter. कैसे? They are fixing atmospheric nitrogen and they are adding organic matter to the soil. If they are adding organic matter to the soil, indirectly what they are doing, they are increasing the fertility of the soil. If the fertility of the soil is increasing, then obviously the plants are getting enough amount of nutrition, they are getting enough amount of nitrogen, and based on that plants are able to grow and their development is taking place properly. This is a whole system or the whole link. Cyanobacteria, they are autotrophic in nature. They fix atmospheric nitrogen. Examples are Anabena, Nostoc and Oscillatoria. They are mostly observed in paddy field and they add organic matter. If they are adding organic matter, they are increasing the fertility of the soil. Indirectly, they are providing nutrition to the plants and the plants are able to grow properly. Their development and growth takes place very precisely without any disturbance. So, here our topic of the whole microbes as biofertilizer is getting completed. As well as with this, we are completing our chapter 10, that is microbes in human welfare. We discussed in the two previous lectures about microbes as a biofertilizers, why we are using them as a biofertilizers. Agriculture yield are increasing day by day. We need more chemical fertilizers. Because of that, day by day pollution is increasing. Because of pollution is increasing, we are facing very harmful effects. Human beings and animals staying on this earth. So instead of that, we have switched to use organic farming. That is, by using microorganisms, we are increasing the fertility of the soil. That are biofertilizers. उसमें से हमने example देखी तो Rhizobium Bacteria, that are rhizomes present in the root of the leguminous plants and providing, taking the atmospheric nitrogen, and they are providing organic form of nitrogen to the plants. Then we have seen example of Activator and Mesospirillum. They are also giving nitrogen to the plants and they are free living in the soil. Then we have seen Mycorrhiza, which are very important for plant growth and development. Why? They are of genus Globus, they are of genus Glomus, and they are present in the root of plants where they provide resistance from pathogens, they provide resistance to drought, and they provide resistance to salinity, that is increase in salt concentration. Then today's lecture we discussed about Cyanobacteria, which are autotrophic. They are preparing their food in such a manner they are also providing organic matter to the soil. So they are increasing the fertility of the soil. Indirectly they are giving proper growth and development to the plants. So here our 10th chapter get completed. Stay tuned for the next lecture, upcoming lecture, where we will be discussing about human reproductive system. So stay tuned and thank you so much.
Friends, like us, plants also require a lot of nitrogen to make proteins. So they absorb nitrogen continuously from the soil, due to which the amount of nitrogen becomes deficient. As you all know, that there is 78% of nitrogen present in our atmosphere, but plants cannot use it in the manner directly as they can use carbon dioxide. They require nitrogen in soluble form. The bacterium called Rhizobium can take atmospheric nitrogen and convert it into a soluble form. These bacteria live in the roots of gram, peas, mung beans, and other legumes and provide them with nitrogen. In return, the plants provide food and shelter to the bacteria. Thus, we can say that Rhizobium shows a symbiotic relationship.
Earth Alive Soil Activator is a certified organic and patented biostimulant. It is composed of a proprietary blend of naturally occurring soil microorganisms and an all-natural forestry byproduct. It does not contain genetically modified organisms or chemically synthesized materials. When applied, preferably during seeding or planting, the beneficial microorganisms of Earth Alive Soil Activator, strains of bacilli and pseudomonads, start multiplying and colonizing the rhizosphere, the narrow region of soil around the roots of the plant. The beneficial microbial population improves the plant's uptake of nutrients by converting the soil minerals into easily absorbable nutrients, thus promoting the plant's health and growth. For example, the proprietary blend of specifically selected microorganisms contained in the Earth Alive Soil Activator transform gaseous nitrogen into a nutrient supporting and enhancing plant growth. It also produces siderophores, high-affinity iron chelating compounds that make iron available to the plant roots. Bacilli are known to produce a wide variety of extracellular enzymes that break down organic matter in the soil, providing a source of nutrients essential to plant growth. Pseudomonads solubilize phosphate contained in the soil, releasing bioavailable phosphorus. Bacilli solubilize silicate and zinc, making it available to the plant. The multiple modes of action offered by Earth Alive Soil Activator lead to healthier plants and higher crop yields, obtained with reduced fertilizer, pesticides, and water use. This results in more sustainable agricultural practices and increased profitability for growers. Earth Alive Soil Activator. Bring life back to life. Visit us at earthalivect.com for more information about our soil activator and our other products.
Cyanobacteria, which are often commonly referred to as blue-green algae, are an entire phylum of eubacteria. They perform photosynthesis and, unlike other bacteria which perform photosynthesis, release oxygen gas as a waste. It was actually the cyanobacteria which first developed this type of photosynthesis about 2.5 billion years ago, and it is largely due to their action that appreciable amounts of oxygen were added to the atmosphere in fossil Earth. Not only are they phototrophs, which can use sunlight to power their metabolic reactions, they are photoautotrophs, which can utilize this sun's energy to fix carbon in organic molecules. In other words, convert the carbon from carbon dioxide, which is an inorganic molecule, into the carbon found in sugars and other biomolecules. As such, they are producers and very important for food chains. Many filter-feeding organisms depend in part on cyanobacteria. Many protists undergo phagocytosis of cyanobacteria. In addition to being producers, which bring biological carbon into food chains, many cyanobacteria can also fix nitrogen. They have the enzyme nitrogenase, which can then take atmospheric nitrogen gas and convert it into more usable forms such as ammonia and nitrates. They are unusual bacteria in that they are larger than normal, and their photosynthesis is performed on internal membranes known as thylakoid membranes, as opposed to other bacteria which perform photosynthesis using mechanisms on the cell membrane. They utilize chlorophyll and other pigments, which is different from the bacteria chlorophyll used by other photosynthetic bacteria. In addition to being important in food chains, they can be symbiotic with a number of separate organisms. They can have relationships with fungi and form lichens. They can form endosymbiotic relationships with certain protists and animals, including sponges, corals to a degree, and other animals. And it was the early cyanobacteria which formed the first chloroplasts in an endosymbiotic relationship in ancestral plants. And so green plants which perform photosynthesis do so because of the endosymbiotic descendants of cyanobacteria which form the chloroplasts in their leaves. Cyanobacteria can be found in almost every environment: freshwater environments, marine environments, and terrestrial environments. And while many are unicellular, they can be colonial and occur in sheets and chains. Some of these colonies can have some degree of specialization of cells, forming, in addition to the vegetative cells performing photosynthesis, specialized aconites, which can withstand harsher environmental conditions and can be the form they overwinter in, and then the heterocysts in some species which allow them to fix nitrogen.
रासायनिक खाद व जैविक खाद। द्वितीय विश्व युद्ध के बाद पश्चिमी देशों के हथियार और विषाक्त रसायन बनाने वाले कारखाने बंद होने के कगार पर पहुंच गए थे, जिसके कारण पश्चिमी देशों को काफी आर्थिक नुकसान हो रहा था। इन देशों ने इस आर्थिक संकट से बचने के लिए एक योजना के तहत उन कारखानों में रासायनिक खाद तथा कीटनाशक बनाना शुरू कर दिया। इन रासायनिक खादों व कीटनाशकों के द्वारा कृषि उत्पादन को बढ़ाने की बात कर हरित क्रांति नाम का एक छद्म जाल बुना। भोले भाले किसान अधिक उत्पादन के लालच में फंसकर रासायनिक खाद व कीटनाशकों का प्रयोग करने लगे। बैलों की जगह ट्रैक्टर से जुताई होने लगी। शुरुआत में कम रासायनिक खादों के प्रयोग से अधिक उत्पादन हुआ। धीरे धीरे रासायनिक खादों के प्रयोग के कारण भूमि की उर्वरा शक्ति क्षीण होने लगी, जिसकी वजह से उत्पादन घटने लगा तथा फसलें कमजोर होने लगी। फसलों के कमजोर होने के कारण उनमें रोग व कीट लगने लगे। रोग व कीटों से फसल की सुरक्षा के लिए और अधिक कीटनाशकों का प्रयोग होने लगा। एक तरफ ज्यादा उत्पादन के लिए अधिक रासायनिक खादों का प्रयोग होने लगा, वहीं दूसरी तरफ फसलों को रोग व कीटों से बचाने के लिए अधिक विषाक्त कीटनाशकों का प्रयोग बढ़ गया, जिसके कारण उत्पादन लागत बढ़ी और किसान को लाभ कम होने लगा, बल्कि छोटे किसान कर्ज में डूबने लगे। धीरे धीरे भूमि की उर्वरा शक्ति क्षीण होने से भूमि बंजर होने लगी। रासायनिक खाद व कीटनाशकों के कारण खाद्यान्न भी विषाक्त हो गए तथा जलवायु भी प्रदूषित हो गई, जिसके कारण मानव पशु पक्षियों के स्वास्थ्य पर प्रतिकूल प्रभाव पड़ा, जिसका परिणाम आपके सामने है। मानव स्वास्थ्य पर रासायनिक खादों के पड़ने वाले प्रभाव को आप ऐसे समझें। जब किसान फसल में रासायनिक खाद डालता है तो रासायनिक खाद में उपस्थित विषाक्त रसायन जल में घुलकर सबसे पहले जल को प्रदूषित कर भूमि के अंदर उपस्थित केचुए व सूक्ष्म जीवाणुओं को मार देता है। ये बात सभी किसान अच्छी तरह से जानते हैं कि केचुए व अन्य सूक्ष्म जीवाणु भूमि की उर्वरा शक्ति को बढ़ाने के लिए अति आवश्यक होते हैं। विषाक्त रसायन के कारण इनकी मृत्यु हो जाने से भूमि की उर्वरा शक्ति नष्ट हो जाती है तथा भूमि कठोर होकर बंजर होने लगती है। रासायनिक खादों के विषाक्त रसायन पानी के माध्यम से अन्य खनिजों के साथ फसल के अंदर पहुंचकर फसलों को विषाक्त बना देते हैं। बाद में जब खाद्यान्न के रूप में मनुष्य या अन्य पशु इन्हें खाते हैं तो ये विषाक्त रसायन मानव स्वास्थ्य पर बहुत ही बुरा प्रभाव डालते हैं तथा मनुष्य के अंदर तरह तरह के रोग उत्पन्न करते हैं। जब हम जैविक खाद का प्रयोग भूमि पर करते हैं तो जैविक खाद पानी में मिलकर पानी को प्रदूषित नहीं करती। जैविक खाद भूमि के अंदर उपस्थित उर्वरा शक्ति को बढ़ाने वाले जीवाणु तथा केचुओं की संख्या को तीव्रता से बढ़ाती है, जिससे भूमि की उर्वरा शक्ति बढ़ती है तथा भूमि छिद्र युक्त एवं भुरभुरी हो जाती है, जिससे वायु और प्रकाश भूमि के अंदर गहराई तक प्रवेश करते हैं, जिसके कारण भूमि में तापमान एवं आर्द्रता का संतुलन बना रहता है और फसलें मजबूत होती हैं। जैविक खाद में विद्यमान तत्व खाद्यान्न के माध्यम से मानव व पशु पक्षियों के शरीर में पहुंचकर स्वास्थ्य लाभ पहुंचाते हैं। जैविक खादों के प्रयोग से फसलें अच्छी और मजबूत होती हैं, जिसके कारण उन पर रोगों एवं कीटों का ज्यादा प्रभाव नहीं पड़ता। यदि किसी प्राकृतिक कारणवश कीटों व रोगों का प्रभाव अगर पड़ता भी है तो गौमूत्र एवं औषधीय वनस्पतियों के घोल के छिड़काव करने से फसलों को रोग एवं कीटों से बचाया जा सकता है। गौमूत्र एवं अन्य प्राकृतिक औषधियों के मिश्रण के छिड़काव से फसलों का कम लागत में बचाव हो जाता है तथा वातावरण पर भी कोई दुष्प्रभाव नहीं पड़ता। यहाँ हम आपको तालिकाबद्ध तरीके से दोनों
Every city or village produces millions of tons of wastewater. Spent water from residences that carry body wastes, excreta, food remains, and laundry wastes, to name a few. Similarly, industries spew commercial wastes through wastewater. All such wastewater is called sewage. Have you ever wondered where all this sewage produced by us goes? It certainly cannot be just deposited as it is into rivers or the sea. Sewage is first treated by microbes that are naturally present inside it before it can be safely disposed of in rivers and streams to avoid polluting these water bodies. That is why the Ministry of Environment and Forests has produced the construction of a large number of sewage treatment plants under the Ganga Action Plan and Yamuna Action Plan to save major rivers like the Ganga and Yamuna. In India, microbes are already treating gallons of wastewater inside several sewage plants around the country every day. Let's take a look at how microbes help in sewage treatment. Microbial treatment of sewage usually takes place in two stages: primary and secondary treatment. In the primary stage, the objective is to remove coarse solids and other large and small particles found in sewage. The first step of primary treatment involves filtration, which is followed by grit removal inside a primary settling tank. All the floating debris is removed by sequential filtration, whereas the grit, such as coarse soil or pebbles, is removed via sedimentation. During sedimentation, most organic and inorganic solids settle, while floating materials like grease and plastic rise to the surface and are skimmed off. The settled solids are called primary sludge, while the floating material comprises primary effluents. Primary effluents are now transferred from the primary settling tank to large aeration tanks for secondary treatment, also known as biological treatment. Secondary treatment aims to reduce organic matter in the effluents. In the aeration tanks, the effluents are constantly agitated mechanically and oxygen is pumped into them. As a result, aerobic bacteria naturally present in the effluents grow into flocks, which are mesh-like structures formed by groups of bacteria in association with fungal hyphae or filaments. In the presence of oxygen, these growing aerobic microbes consume the organic matter present in the effluents. This secondary treatment reduces the BOD or biochemical oxygen demand of water. BOD is an important measure of water quality. It refers to the amount of oxygen needed by bacteria and other microbes to oxidize all the organic matter in 1 liter of water. The BOD of drinking water should be less than 0.5. However, the BOD of raw sewage can be as high as 600 milligrams per liter. That is, the greater the BOD of wastewater, the more polluted it is. Also, it is an indicator of the amount of organic matter present in water. Secondary treatment is continued till BOD decreases considerably. Once this is accomplished, the effluents are transferred to another aeration tank where the flocks of bacteria are gradually allowed to sediment. This sedimented part of sewage is known as activated sludge, which is pumped into large tanks called anaerobic sludge digesters. Here, anaerobic bacteria digest the bacteria and fungi present in the activated sludge and produce a mixture of gases such as methane, hydrogen sulfide, and carbon dioxide. This mixture of gases is also called biogas and is used as fuel since it is inflammable. Methane is the main gas produced from sewage treatment and also serves as fuel. It is used to produce heat in homes and factories. The sludge left after digestion of both aerobic and anaerobic bacteria is used as manure and is called activated sludge. The effluents that now remain from the secondary treatment plant are now fit to be released into natural water bodies like rivers and streams. However, a small part of the activated sludge is pumped back into the aeration tank to serve as inoculums for subsequent treatments. Though the amount of sewage produced increases every year with the population increase and rapid urbanization, microbial treatment of sewage remains the most effective method to date.